Hello, my name is Emily Vengelo, and today I will be talking about Colorado tick fever. So Colorado tick fever is caused by a virus, formerly known as Colorado tick fever virus. Its common names are mountain fever, mountain tick fever, and CTFP. It belongs to the genus Cultivirus, the family Rheoviridae. The vector of transmission is an arthropod known as the Rocky Mountain wood tick, Dermacentor andersoni, found in the western U.S. and Canada between 4,000 and 10,000 feet above sea level and grassy areas near sagebrush. Here is a picture of the female wood tick and the male wood tick. Female wood tick has cream-colored scutum. Male wood tick just same color throughout. Here's an electron microscope image of the Colorado tick virus. Taxonomy, like I said, it belongs to the family Rheoviridae. It also belongs to the subfamily Spina Rheovirinae, and the genus is Cultivirus. There are other cultiviruses that are related to CTFE, one being CTFE California, which was a species that was isolated from a white hare in California. Then there's also the Salmon River virus. So these three are found in North America. If you look across along the same latitude, you'll find what's known as the EAC virus in Germany and France. And this is not evolutionarily similar to these, though it is serologically, so antigenically related to these guys over here. And so it does have similar symptoms to CTFE. It actually results in neurological complications but more importantly, febrile illness. But the difference is that I guess it's spread by exoded ticks. Here's a funny, a confusing little case. They don't really know how it happened, but there was a human in South Korea in the 1960s who contracted CTFE, or they found the presence of CTFE antigen in his serum, and they don't know how he got it because it's not typically, CTFE is commonly found in North America and not here. So maybe he traveled there. We don't know. Okay, moving on to characteristics of this agent. So it is a non-enveloped virus, icosahedral, has a triple capsid structure. So you can see it in here too, but like the black ring is the outer capsid, gray ring, intermediate capsid, and then the white ring is the inner capsid. Within that is the naked core, and that is where the genome is found. Viral genome, class 3 on the Baltimore classification system, double-stranded RNA, has 12 segments of RNA. You can see it's separated out here in electrophoresis. And then this just indicates that it's 100, this white bar is 100 nanometers, so the diameter is 60 to 80 nanometers. Something like that. Moving on to notable strains, I did talk about CTFE California strain. I want to talk about the Florio strain. It is the very first strain of CTFE isolated. It was isolated from humans and then injected into mice and discovered as the causative agent of Colorado tick fever. And Florio was the one who discovered that. And so he got to be named after, the strain was named after him. So later in 1987, a study was done, and at least 22 strains were known at the time, though the antigen, like the variation in antigens, is very low, like it's basically the same. So this research study actually was used to, I guess, inject the different strains into mice and see, like, you know, the differences. And so Florio was actually the only strain that caused deaths in 3 to 4 week old mice, as well as like earlier death time. So at least 2 days earlier than all the other strains. So it was it's a highly pathogenic strain. CTFE California strain, right here. It was isolated from the white hare known as Lepus californicus, and even though it's like in California, outside of the range of Dermacentor andersoni, which is the causative agent of CTFE, it's interesting to note because even though they're geographically located like they're close, and so you can kind of see how they would evolve together, and they're closely related. And then EAC virus, as I said, is like distinctly evolutionarily not related, though close. Moving on to pathogenesis. So this virus actually infects our erythrocyte progenitor cells in the bone marrow. And so once your erythrocytes mature and develop, the virus will be present inside those erythrocytes until the cells die at about 120 days or 4 months. So the virus, it enters through our cells. It first has to recognize the cell receptors, and a bit of encapsulating or Uncoding of the capsid occurs in double-stranded RNA unwinds, positive single-stranded RNA is synthesized, then translated to form viral proteins. Then this is transcribed to form negative single-stranded RNA. Bases are added by polymerases to form the additional strand, which will bind together. And then the capsid will be assembled around this newly formed double-stranded RNA. And then it will exit out the cell and cause cell lysis, and be known as a mature virion. So, incidence and prevalence. So this disease occurs in the Rocky Mountain area where the Rocky Mountain wood tick is found. So people who visit, travel, or work in these areas are more likely to get this disease, most likely to get this disease if they encounter or are bitten by an infected Rocky Mountain wood tick. It is not a leading cause of death, and it's endemic to West US and Canada between 4,000 and 10,000 feet above sea level. Most cases occur during the spring or summer months when ticks are most active. And as of 2010 to 2019, only 59 CTF cases have been reported. So it's not a nationally reported disease, but only in the states where this disease is prevalent. And I've listed the diseases, I've listed the states where the disease is prevalent here. Moving on to the history. So it's not a new virus. It's possible that Lewis and Clark. So Clark had suffered from CTFE, ColoradoTickFever in June of 1805. Dermacentor andersoni was identified in 1908 by Charles Stiles of the U.S. Public Health Service. Andersoni was named after J.E.F. Anderson, who hypothesized that Rocky Mountain spotted fever was transmitted by this same wood tick. Sometimes people can confuse Rocky Mountain spotted fever with Colorado tick fever. Rocky Mountain spotted fever is a bacterial. It's caused by a bacteria known as Rickettsia rickettsii, and then Colorado tick fever is caused by a virus, ColoradoTickFever virus, and that's what we're talking about. So in 1930, Becker actually described and coined the term ColoradoTickFever virus. He, based on observations from Colorado, so you get ColoradoTickFever virus, he noticed that this virus, there wasn't that iconic spotted rash that you see in Rocky Mountain spotted fever, and there was also a higher overall recovery rate. And in 1945, Florio and his associates at the University Medical Center in Denver, Colorado, were able to recognize that CTFE is the cause of human disease and that the vector for it is Dermacentor andersoni. Moving on to outbreaks. So there's two outbreaks that I have listed here, one in the past, one more present. 1980 to 1988, there were about more than 1,400 cases within a decade. So, and most of those cases were in Colorado. But nowadays, we don't see that many cases. We see, like, remember the previous statistic was 2010 to 2019. It's like only around 50 cases. And another outbreak occurred in Wyoming last year, July of 2020. They typically report 0 to 2 cases a year, but this year they reported 5 cases, so that was concern for outbreak. Some two recent events that I'd like to compare to help us understand CTFE. One occurred in April 2017, one in summer of 2018, so both in the summer. One, there was a 65-year-old woman hiking in Canada. The other, there were 4 cases in Oregon. These people lived in 3 close-by counties, and both of these cases, they were bit by tick. They had a first period symptoms were high fever, illness, chills, myalgia, reduced appetite. They had a period where symptoms subsided, and then a second period, part of the biphasic illness, where those symptoms recurred, and they had fever, illness, chills, headache, reduced appetite. What was common also among these 2 cases is that there was significant leukopenia. So there was significant decreased neutrophils, monocytes, lymphocytes, platelets, and that makes sense because the virus actually targets our erythrocyte progenitor cells. So that's how you can see that there would be a decrease in these in their blood tests. In both cases, there were some who were hospitalized, but all made 100% recovery and no ongoing symptoms after that. And the CDC was involved with both, diagnosed the virus in these patients through RT-PCR, reverse transcriptase polymerase chain reaction. Transmission spread to people or animals through the bite of infected Rocky Mountain wood tick, Dermacentor andersoni. There's been a rare case of blood transfusion from person to person, but that's very rare. CDC recommends you don't be a blood donor or bone marrow transplant donor. until like 6 months, for at least 6 months after you get the virus. And then, so how does this actually spread? So the reservoirs for this is the small rodents known as squirrels and chipmunks and mice, who are the reservoirs for this virus, and then larvae feed on this reservoir, develops into nymph, then into adult. The infected adult tick feeds onto either a human or other animals, so like deer, elk, and so then these become infected because the tick actually bites down through port of entry being the skin, and then the saliva contains the virus, and it just enters your bloodstream. At-risk populations include anyone who's been bit by a tick in an area where CTF virus is spreading, so basically anyone. Rocky Mountain area, again above 5,000 feet, forest, brush, or wooded areas, especially in those summer months when ticks are most active. People are living, traveling, hiking, camping in those areas. Prevention, there's no vaccine, so just avoid tick-infested areas, especially during those warmer months. Wear light-colored clothing so you can easily see, if ticks are crawling on you, you can move them. Tuck your pants into your socks and wear long-sleeved shirts like in chem lab, just cover up. Tick repellents, you can use tick repellents that are EPA registered, because these tick repellents, they kind of provide a slippery surface on your skin so that the ticks can't actually bind or can't latch onto your skin. Also, regularly inspect your ticks. Regularly inspect your body for ticks. Check the crevices where they like to hide, and you can see in these pictures. And then if you do get a tick bite, use tweezers to remove the tick as close to your skin as possible and pull up. And don't squeeze the tick, because otherwise you might squeeze its fluids into your bloodstream, which will further increase your chances of contracting CTFE. And then also after that, you would wash the infection site with blood and water. And so about the pathology, so like I discussed, so it infects our hematopoietic progenitor cells in the bone marrow, and so as a result, you'll see a decrease in all these cells, and it's nonspecific. Immune response, so incubation period occurs about 1 to 14 days from the time of the tick bite. The virus replicates in our erythrocyte precursors, so our cells undergo humoral response. The virus specifically targets CD34 plus antigens, which are on the hematopoietic cells. The virus forms, granular matrices and filaments, and here the virus is entering and through endocytosis into the erythroblast. You can kind of see the membrane is being pinched in, and neutralizing antibodies are only formed 2 to 3 weeks after the onset. And so, but the thing is, even though there's neutralizing antibodies in the bloodstream, the virus is protected from these antibodies because it's like hiding inside the blood cells. So even though the neutralizing antibodies are like flowing around the blood cells, like the virus is not being neutralized, and it's a sneaky little guy, I guess. Here are the symptoms. I talked about this. You can look at it if you'd like. Diagnosis, there's 2 main ways: RT-PCR, reverse transcriptase polymerase, and serology to detect those neutralizing antibodies. But again, like those show up 14 to 21 days after, so it's not commonly used. RT-PCR is quick and efficient. You can detect it within 5 days to see if you have CTFE. And then we talked about that. Okay. Treatment, there's no treatment. Just take acetaminophen to relieve your symptoms of fever and pain. Up to 30% of people will require hospitalization, but they just get supportive care like IV fluids. Complications are extremely rare, like aseptic meningitis, encephalitis, hemorrhagic fever. Don't take aspirin because that could worsen hemorrhage. Once infected, you will have immunity against reinfection. Individuals above 30 will have symptoms that may last longer, and children are more often prone to neurological complications and mortality. Research efforts, 1993, permethrin on cotton was used to kill rodents that were infected with teas. This was successful, and so you'll see permethrin and EPA tick repellents to like spray on your gear and clothing and stuff. Ribavirin was considered possible treatment, but it wasn't really tested on humans, so that kind of failed. In 1960, there was an experimental vaccine for tick-borne encephalitis in Russia, but that did show long-term immunity, but they discontinued it. In 2020, the CDC announced a national plan for identifying where tick populations are increasing why, and so that is what the future will look like, is just prevention. And so here are my resources, and if I have time, I'm going to go back. So yeah, just prevent tick Take bites, follow what the CDC recommends, like what we talked about.